Hello everyone, and welcome to Pearson Bio Academy. I am Fazna Imran.
This is the place where you can top up your biology knowledge with our past paper explanation through the very clear and short video series provided by us. And we are currently focusing on Pearson Edexcel International Advanced Level, paper reference WBI 1201 and Unit 2, which covers cells, development, biodiversity and conservation. Let's get started. If you have watched my introduction video, then you might know there are 6 key topics under Unit 2 and currently we are discussing past paper questions on cell structure. And these are the categorization I have made for you to make it easy. And today we'll be focusing on types of microscope and staining. This is my personal tracking system which I used during my A-level studies. It's really important, especially when you are learning past paper questions or when you want to learn anything new. It's better to have a section tracker. So these things will help to keep your lessons organized and also your brain on the track. So you will be more coordinated. And also you always know what you have learned already and what you want to learn in next. And this method is 100% effective based on my self experience. And in last video, we discussed questions about magnification from 2019 to 2024 papers. And if you have not watched my video yet, I'll put the link in the description box. Please check it out. And today we are going to discuss questions about types of microscopes and staining. Let's get started. Under types of microscope, you will often have questions that check whether you have a thorough understanding of using microscopes. You have got the light microscope and the electron microscope. So sometimes the examiner can give you a picture and test if you can identify which microscope should be used for that particular image. Now, I know some of you are following school and some may not be, but many of you might have experience using a light microscope even at your small classes. I think so. And as a medical laboratory scientist, to be honest, I have never used an electron microscope. It's mainly because it's quite expensive and also it requires a lot of training. And on the other hand, a light microscope is much simpler to use. So the goal of this type of question is to see if you know when to use each microscope correctly. So the key focus is understanding which microscope is best suited for different situations. So first, when you are comparing the two microscopes, there are two key terms you should know. The first one is resolution power. So what is resolution power? Well, let us say if you think about buying a camera and you look for good pixels and zooming capabilities, right? So similarly, in a microscope, you need to consider certain features and resolution power is the most important out of them. So what does resolution power mean? A measure of how close together two objects must be before they are seen as one. And now let's look at the two microscopes, the light microscope and the electron microscope. And the resolution power of light microscope is something around 200 nanometers. And for electron microscope, it's much higher. It's something around 0.1 to 1 nanometers. Let me explain like this. If you have 2 points, point A and B, and a light microscope can only distinguish them if they are at least 200 nanometer apart. But electron microscope can distinguish points that are as close as 0.1 to 1 nanometers. So electron microscope can reveal much fine details compared to a light microscope. And why does this matter? Let us say if you are looking at a chloroplast under electron microscope, you will see a highly detailed image. The inner structure like the membrane, starch molecules are shown very clearly here, and you can observe the fine structures very easily. So this is because the electron microscope has both high resolution power and high magnification power. So it allows you to enlarge the image and distinguish even the smallest detail between 2 points. But if you look at the same chloroplast under a light microscope, let us say this is a cell from a plant, and this is how the chloroplast look like. You will only see a general wave like some particles, but not the fine details. So if you need to see those inner structures or want more precise details, you would definitely need to use an electron microscope. So the next key feature is magnification. So magnification means how many times that specific microscope can enlarge the given object. And for light microscope, it's about 1500 times, and for electron microscope, it can magnify an object up to 500,000 times. So you can see as resolution power, the magnification power also much higher in electron microscope. And we are moving to the first question from 2019 June paper. So this question about pancreas. So a student observed some cells of the pancreas using an electron microscope. So they have mentioned clearly which type of microscope they have used. And this is how that organelle looks like. Several curved membrane bound sacs of decreasing size. So when I draw it, it looks like this. So most likely this could be Golgi apparatus. And this question was explain why this organelle cannot be seen using a light microscope. So when you are asking to do some explanation, then you have to give some justification, or else you have to give a proper reason why you can't see this organelle by using a light microscope. So basically this Golgi apparatus are very small structures, and due to the limited resolution power and magnification power of light microscope, you would not be able to see the structures very clear using light microscope. So the question is very easy, so you can give your answer like this. The resolution power and magnification of a light microscope is not enough to see this organelle because this organelle is very small. So the next question from 2019 October paper, you are given with two different pictures. One is cell seen using a light microscope, and the next one a part of cell seen using an electron microscope, and they are focusing on cell surface membrane. So if you look at electron microscope, you can see the bilayer structure which are distinguished from the other. But in light microscope, you cannot see that bilayer distinction, just they are showing as one layer. And the statement was the bilayer structure of the cell surface membrane can be seen only with an electron microscope. So here you can see that these two layers are differentiated from one to the other very clearly. And your question is, explain why the bilayer structure of the cell surface membrane could not be seen using a light microscope. I think you're almost on the way to your answer. So this question is about why you can see the bilayer structure clearly. So here you can't talk about the magnification because you know when you want to distinguish 2 points, then that microscope should have high resolution power. So here, if you're talking about magnification or if you're explaining your answer using magnification, that it would be completely wrong. So what was your point? Yeah, the electron microscope has a high resolution power, or else you can say in conversely, the light microscope have a very low resolution power. That's why you can't see the bilayer structure very clearly through the light microscope. You can answer simply like this. The bilayer of the cell surface membrane is too close together for a light microscope to resolve due to its low resolution power, making it appear as a single structure rather than two layers. The next question from 2020 January paper. Here also you are given with a picture, and if you are familiar with the organelles, then you know this is a picture of mitochondria. And this question is not about the types of microscope which you use to view this one, but it's related to the microscopical observation. That's why I bring these questions under here. And in this question, they have stated that T and U are the same type of organelle, but which looks different. And the question is, suggest why these structures look different in this photograph. So if your command word is suggest, then you know you have to think out of the box and you have to come up with the logical answers. So these questions are not directly from your theory. You have to think and write. So normally we know the mitochondria have the rod shaped, or they have the shape of sausages. So you have that shape, but T is having a circular shape. So please keep remember the cells in real scenario, they are spherical. So they have the 3D structures. So when they focus these things, they could be viewed in different angles. So one can be in transverse and another can be in longitudinal way. That's why, therefore, these two can be look differ. And also, you know, before focusing on the microscope, the samples has to be prepared. And during the process, the cells can be cut off. And during that, the mitochondria can be sliced off. So therefore also you can see these different in their shape. And apart from that, you can include these third picture also. You can see three of them were looking in different sizes. So they can suggest you that they might be in different phases of their growing stage. So these are the suggestions you can make. They can be viewed from a different angle where one is viewed transverse and the other is longitudinal. And it can be viewed after cell cutting where a mitochondria has been sliced. There can be presence of different sizes of mitochondria due to varying stages of growth or development. The next question is quite easy. Here also you are given with a picture of mitochondria again with a magnification of 72,500 times, and the question is you have to state which type of microscope had been used to view this one. And due to the detailed image plus high magnification, it should be an electron microscope. The next question is from 2021 June paper. This question is quite similar to the previous one, but little bit modified. So here again you have given with a picture of bacteria. And first you have to find out the magnification, but in the previous one they have already given you the magnification. And then you have to explain which type of microscope you can use for this purpose. So first, if we find out the magnification, it's around 29,000, which is very high. So definitely it should be an electron microscope because it only allows this much of magnification. So you can't use light microscope. And apart from that, this image, however, it appears like 3D. So you can use the specialized electron microscope, scanning electron microscope here for view some 3D structures. We will be moving to the next question. It's from 2022 January paper and it's about a bacteria called Listeria monocytogenes, which are very small in size and they are around 0.5 micrometer in length. And in this question, they have asked you to find out the magnification first. So you can find out the magnification through this and it's about 16,000 times. And the question is, state why a light microscope cannot be used. So here they ask you to state. So it would be enough if you give the reason only, therefore you don't need to give any explanation. And definitely you can't reach that much of higher magnification using a light microscope. So therefore you have to use an electron microscope. And additionally, if you look at the size of this microorganism, they are very tiny. It's about 0.5 micrometer, which is equivalent to 500 nanometers, which is very close to the resolution power of light microscope. Therefore, they cannot give very detailed, clear image. And most of the time, the image can be blurred. So you can give the answer like this. The resolution power and magnification of a light microscope is not enough to see these bacteria because these bacteria are too small. And if you have write anything referring to the 3D image structures, then you will not gain any marks. The next question from 2023 January paper from a recent year, you can see this one. So first, the photograph A shows some cells in one type of algae as seen using one type of microscope. So here you can see they are focusing on chloroplast, which looks like some particles. And the photograph B shows the same structure as seen using a different types of microscope. And I think you almost guessed what type of microscope used for focused photograph A and what would be for the photograph B. And here, as the previous one, they first ask you to find out the magnification. Then the question is, explain why the microscope used for photograph B shows more detail than the microscope used for photograph A. And here we have to talk about the resolution power. Here you can't use the magnification. The electron microscope was used for photograph B, therefore it allows to distinguish between 2 points that are very close together, showing finer details due to the high resolution of electron microscope. And I think this question is similar to that bilayer structure, and that question also they gave you two pictures from focused under light microscope and the other focused under electron microscope. And the second picture give you a very detailed structure, then they ask you the reason, so only you have to talk about the resolution power. And the next question is from 2024 January. And again, they have given a picture of chloroplast. And with this fine detailed structure, you can identify that it has been focused using an electron microscope. And the question is, name the type of microscope, and you have to provide one reason for your answer. So it's quite simple. If you write electron microscope, then your reason could be because of high resolution power. And also you can write this one, the transmission electron microscope, because this will allow the ultrastructure of the chloroplast to be seen, or they could produce very detailed image. Therefore you can use this one, but you cannot use these terms like image is clear, or because of this high magnification you can't provide these things. I think in previous question also I talked about this. So that wraps up our discussion on the types of microscope. I hope the explanations were clear. We have covered questions from papers spanning 2019 to 2024. Now let's move on to the next section, which focuses on staining. And staining is closely related to the use of light microscopes, and most of the time we stain cells, tissues or particles to enhance visibility under the light microscope. And it makes it easier to distinguish and identify specific structures within a sample. For example, you can give the starch molecules were stained by iodine, then you can identify that starch storage very easily. And if you have done lab classes, you may have performed one or two staining procedures by yourself. And you know, the histologist like us, we prepare tissue samples for microscopic examination, and we frequently use staining techniques. And these procedures help produce clearer and more detailed images, allowing for better identification and analysis of the samples. Before moving to the questions, we will quickly go through the common stains which are available. And these facts I have driven from your student book, not anywhere else. And very first we have hematoxylin, which stains the nuclei of plant and animal cells into purple, blue or brown. Then we have methylene blue, which stains the nuclei of animal cells into blue color. You know, the hematologist who deal with blood samples, often they use methylene blue to stain the patient's blood smear. So then it will give you some nice pictures with detailed differentiation among blood cells. And then we have acetocarmine, which can stain the chromosome in dividing nuclei in both plant and animal cells. You know, if you want to find out the mitotic index, then you have to make differentiate from the cells which undergoes mitosis and the cells which don't. So in that purpose, you have to make the clear distinction in between them. Therefore, these acetocarmine can be very helpful. And you know, like in most frequently they are asking questions related to this stain, but they have never asked you to write the exact type of stain which used for this purpose. They are asking about what are the advantages of using this stain, but maybe in future they could ask. And finally, we have iodine stain. You know, from the beginning, we have learned iodine can stain the starch molecules. And if you find out any starch containing materials in plant cells, then simply you can use iodine. And we have the first question from 2020. So it's about a pea plant which have some tendrils. And your question is, a tendril is a modified leaf that can coil around anything it touches. Cells in the tip of the tendril divide by mitosis. A student investigated the mitotic index of cells in the tip of tendrils. An acetic ozone stain was adhered to the tissues from the tips of the tendrils. Give one reason why the cells were stained in this investigation. So the answer is to make the chromatids or chromosomes or DNA to be visible. You have to specify with your answer. You have to say which structure is stained by that one. The next question is from 2023 January paper. It's about a blastocyst. The blastocyst cell was treated to show the stage of mitosis, and the question is state how the blastocyst cell was treated to show the stage of mitosis. So if you want to see these chromatids very clearly, then you have to go for a staining. The answer is a stain was applied to the cell to make visible one of them. You have to be very specific with your answer. You have to say which structure you have to stain. It can be either DNA, chromosome, chromatid or histone protein. And also in this answer, you can see that the examiner didn't expect you to write the correct type of stain. Instead of that, writing as stain would be enough here. The next question is from 2024 January paper. Here a sample of tissue was taken from a patient with this type of cancer. Previously they have explained about the cancer in order to calculate the mitotic index. This sample was stained before being viewed using a microscope, and the question is state why the cells in this sample were stained before being viewed using a microscope in order to calculate the mitotic index. So it's very easy to make the chromosomes or chromatids visible under the microscope in order to distinguish which cells undergoing mitosis or to identify the cells in mitotic phase.
And don't forget to like, subscribe and follow the Pearson Bio Academy channel for more continuous learning and exciting new videos every week. Stay tuned.